So as I said, we are now going to discuss about how the fertilization takes place. So if you see the fertilization, we just saw about the pollen grain. So pollen grain was this way. This was the pollen grain, and it had got the outer kind of a exine and the inner one which is intine, and then there is this particular kind of a thing. So this, when it is going to develop, the nucleus over here is going to have, one is the tube nucleus and there is the generative nucleus, and they are going to form a tube. So there will be a tube which is going to be formed, and this tube will be having three nucleus inside. So the generative nucleus getting divided into two sperm nuclei, and the tube nucleus is going to produce the pollen tube inside the style of the flower. Now when we see the style and the stigma over here, so we can see the stigma will be something like this, on which there is this particular pollen grain. The stigma is going to have a style which will be like this over here, and then to the bottom will be the ovary. So the ovary is in this particular form. Inside this ovary there will be this embryo sac which will be something like this. The embryo sac will be over here, and this embryo sac, this will be the nucellus. So this is the nucellus, and this one is the embryo sac. Okay, and now inside this will be some different kinds of cells. So 3 cells here, 3 cells over here, and 2 nuclei over here. Okay, these ones over here are called as antipodal cells. Okay, these are called as antipodal cells. These two which are over here are called as the polar nuclei, and these ones over here are the middle one is the egg cell, whereas the outer ones are the synergens. Now of course this is the style and this is the stigma. Now what actually is going to happen is this pollen grain is going to start developing a tube like this, and this tube is going to enter inside the style, and it is going to enter into this particular ovule through a opening which is called as micropyle. So inside the embryo sac, it is going to enter inside the embryo sac using micropyle. Now so this 3 nuclei which are there, this is the tube nucleus which is forming the pollen tube, and these 2 are the sperm nuclei. So this tube nucleus is going to disintegrate as soon as it reaches this point, and then what is going to remain are only 2 male nuclei. The male nuclei which are there, the sperm nuclei which are there, they are going to retain over here when they reach here. The tube nucleus is going to disintegrate because उसका काम क्या था? उसका काम था एक pathway बनाना, एक रास्ता बनाना for the pollen grains or you can say the generative nucleus to go into the style and reach the ovary. So you can see over here about the when we talk about the embryo, or we can talk about the ovule, you find that there could be a single ovule producing a single. So there may be just one particular ovule present which will be which will be forming a single seeded fruit. So a fruit with having on a single seed will be one, or else it could happen that it may have multiple ovules and all of them getting fertilized, and that's why it will be a fruit which will be having multiple seeds inside. Now the ovary which is there, the outside part is there, it is going to form the fruit, and the ovule will form the seed of the fruit. So integument leave a small opening called as the ovule is protected by the integument. So the integument are outside, the outside are called the integuments, and they have got a small opening over here which is called as the micropyle. So micropyle is the one through which the pollen tube enters into the ovule. Enclosed by the integument is the nucleus, a mass of food-laden cells, and further inside the nucleus is the embryo sac. The embryo sac consists of 7 cells. There are 7 cells in this embryo sac. Okay. What are those? 3 cells at the microbial end, 1 egg cell and 2 synergids. So 1 egg cell and 2 synergids. There are the 3 which are on the microbial set, on the side where the microbial is there. At that point, there are 3 cells. One is the egg cell, and there are 2 ones which are synergids. 3 cells on the opposite side, which are called as the antipodal cells. The antipodal cells which are there on the opposite sides of to this one are the 3 antipodal cells. And then 1 large central cell. The central cell contains 2 nuclei called as the polar nucleus. So this thing is a single cell. This is actually a single cell having 2 nuclei in it. Okay. The 2 nuclei which are called as the polar nuclei. So 1 central cell which contains 2 nuclei called as the polar nuclei fused together. Now let's see how does the germination of a pollen grain takes place. So let's see what exactly happens over here. As I told you already, that what is going to happen is, once the conditions are favorable, the tube nucleus will start generating a tube from the intine, and it is going to travel downwards into the style to enter into the ovule. So when it does so, meanwhile, the generative nucleus which is present in the pollen grain is going to divide into 2 main nuclei called as the sperm nuclei, and it is going to come over here to enter into this particular ovule for fertilization. So let's see what exactly happens over here. Pollen grain germinates only if it falls on the stigma of the same plant species, otherwise it disintegrates. Yes, in case it is of a different kind of a plant, that the species is different, then it is not going to allow this thing to generate. So it is just going to disintegrate and will not allow any fertilization to take place. The pollen grain, on falling on the stigma, is stimulated to germinate due to the secretion of sugars by the stigma. So there will be certain amount of sugars which will be generated by the stigma over here, which is giving the pollen grain the, or we can say that, ठीक है, password दे दिया हम लोग ने, एक kind of a communication that okay, you are of the same species, so you can continue your work. So this is how the germination is going to take place, by the secretion of sugar by the stigma when it is of the same species. Through a point in the axene, the pollen tube grows out of the pollen grain, carrying at its tip the generative nucleus and the tube nucleus. Generative nucleus divides into 2 sperm nuclei, also called as male gamete nuclei. Thus, in a germinating pollen grain, there are 3 nuclei which are not separated by cell walls. They share a common cytoplasm. So, in the same cytoplasm, in the same kind of a cell, there will be 3 kinds of nuclei, 1 tube nuclei and 2 sperm nuclei. That is a gamete nuclei. The pollen tube grows through the stigma and style by dissolving the tissues with the help of enzymes and reaches the ovary. There it pushes through the micropyle and reaches the embryo sac. The tube nucleus, which had directed the growth of the pollen tube all the way down, now disintegrates. So it is going to keep on digging inside over here. The tube nucleus is going to keep on digging inside the style, and due to its weight, dissolving the tissue, it is going to make a pathway for the cell to reach the micropyle over here. So when it reaches the micropyle, as soon as it reaches the micropyle, the tube nuclei disintegrates. Now the pollen tube enters one of the synergids and releases its 2 sperm nuclei. Of these, 1 sperm nuclei fuses with the egg cell nucleus to form the zygote, while the other sperm nucleus moves towards the 2 polar nuclei in the central cell and fuses with them. Thus, 3 nucleus fuse together, triple fusion, to produce the endosperm nucleus. So you can see what happens is, the 2 nucleus comes over here, enters into the through the synergids. One of the nucleus is going to combine with this one. So there will be this particular form, egg cell with 1 sperm nuclei giving you the zygote. So this forms the zygote. Okay, the main baby. Whereas the other nucleus goes and enters over here and fuses with the polar nuclei. Here, 3 nuclei are going to fuse together, and that is why this one over here is called as triple fusion. Okay, why triple fusion? Because 3 nuclei are going to fuse together. That's why it is called as a triple fusion. Altogether, 2 fertilization have occurred, and hence the whole process is called as double fertilization. So you can see that 2 sperm nuclei fused with 1 egg nuclei and other with the polar nuclei. Accordingly, there are 2 fertilizations taking place. Because there are 2 fertilizations taking place, it is called as double fertilization. So one thing is between egg cell and the sperm nuclei to fuse with the zygote, that is one fertilization, and second is polar nuclei combining with the another gamete nuclei, male nuclei, to give you the triple fusion to form the endosperm. So in this way, endospermic nucleus is going to be formed and double fertilization takes place. So double fertilization, one sperm nucleus fuses with the egg cell nucleus and another sperm nuclei fuses with the 2 polar nuclei of the central cell. So this is how the double fertilization takes place. You can also see that in the figure part, just near the diagram, they are given the details about the 2 fertilization. So 2 polar nuclei unite to form secondary nucleus, and then 1 sperm nucleus from the pollen tube unites with the secondary nucleus to form endosperm cell. This is described as triple fusion involving 2 polar nuclei and 1 sperm nuclei, that is 3 nuclei fusion. That is 1 fertilization. And second is the other sperm nucleus enters the egg cell to the assistance of synergids, which then disintegrate and unites with the nucleus with second fertilization. That's why the example over here gives you the 2 double fertilization and triple fusion both together. So that was about how the process of the fertilization takes place. So we saw that it's very clear. This diagram very important to draw as well as to label. So yes, you need to draw this figure also. There is a full possibility that they can ask you to draw the figure of the double fertilization or the pathway, or you can say just germination of pollen for germination, for fertilization. Now we see that what happens to the floral parts of that particular flower after the fertilization has taken place. So okay, we have seen that the fertilization has taken place. So now we form that the zygote is formed over here and the endosperm is formed over here by this particular thing. Now what else? What exactly happens to that particular flower now? So after fertilization, the flower has served its purpose. So the work of flower for the plant is over. The plant has completed the flower has completed its job of fertilization, and is producing a baby seed which is there in the plant as a zygote. So the petals, stamen, style, stigma wither and generally fall off. So all the 4 whorls, the overall part, that is the petals, stamen, style, stigma, जो भी ऊपर का जो part है वो सब wither off. They are just going to wither off, fall off. Okay. The calyx may either fall off or may remain intact in a dried or shriveled form. Apple and guava clearly show such dried sepals. In brinjal, green calyx persists and remains attached to the ripe fruit. So you can see that in apple also, apple जो वहाँ पे लटकता तो नीचे की side पे you will find that leafy kind of a thing, grey brown color का. Even in case of guava, we find that thing. So what is that? That is nothing but the original calyx of that particular flower. So whereas in brinjal, the upper green color part of the brinjal is nothing but the calyx of the original flower. So that's how it is. Certain times the calyx is going to be retained by the plant, or it is going to be wither off like any other part. The ovary enlarges to form fruit. The ovarian wall forming the fruit wall. The ovary wall may either form a dry or a hard fruit wall or a fleshy fruit wall. So either thing is that, so this particularly ovary wall which is there, that is going to form the fruit wall. Now either thing is the fruit wall will be fleshy. So fleshy agar rahega to we can eat that particular fruit, or it may be hard like a grain, So if it is a grain sort of thing, then it will be hard coating on top of it. So depending upon the different kind of plants, the ovary wall may either be dry and hard, or it could be fleshy. And the ovules which are here becomes the seed. So the ovule, there are plenty of ovules in the flower, maybe one or maybe multiple. So if it is multiple, then these multiple ovules become the seed. The fruit contains one or more seed which are developed from one or more ovules attached to the ovary by means of placentate. When the fruit is ripe, the seeds contained inside are released by one or the other method and grow into new plant in suitable conditions. So yes, so wherever the seeds which are there, they are going to develop and they are going to be one or more ovules agar rahenge, so they are going to form into a seed and are attached to the fruit in the form of its placentation. And when the fruit is ripe, the seeds will contained inside are released by one or the other method, like you are eating it and you are throwing the seed away. So that is one kind of a dispersion of seed. Or maybe the fruit will be matured on the plant itself, and it is ruptured, and then the seed is going to be dispersed. So there are different methods of seed dispersal which would take place. And when the seed is going to get the conditions which are favorable for germination, they are going to grow into a new plant. So the fate of the various part of the ovary after fertilization during the formation of seed is as follows. Ovary forms the fruit. So the ovary forms the fruit. Ovary wall is the pericarp. Pericarp is the outermost part, that is the pericarp is the ovary wall. Ovule forms the seed. Placenta is the stalk of the seed. Outer integument and inner integument are the seed coats. So the integument which were there, they are forming the seed coat, that is the testa and the tegmen. Secondary nucleus, the secondary nucleus which was here, that is over here, this is forming the endosperm, and excell and synergens form the embryo, and antibodial cells, they disorganize. So this depending upon plant to plant, it is going to be disorganized. So that is what happens to the parts of the flower after they are fertilized. So this was about the chapter, quite an easy chapter, because self-pollination, cross-pollination and the different kinds of agents, बहुत simple है. The only thing what is important is this particular part which is slightly new to you. Apart from that, there is nothing difficult over here. Right? You need to understand, you need to write or properly and be able to write about the double fusion and the triple fusion and the double fertilization, and the diagrams also are important in this particular part. Okay, so that was about the chapter pollination and fertilization.